this video, we'll discuss the process of protein synthesis to somehow give clarification on the gray areas on our topic. Generally, we have two major processes in protein synthesis. We have the transcription that happens inside the nucleus, and we have translation phase, which normally happen in the cytoplasm of the cell. Imagine that this is a whole area, an inner area of our cell. Let's start with the transcription process. Now, protein synthesis will not start without the transcription process from DNA to RNA. This portion of the DNA, as you can see, is now unzipped. So you cannot see here a space which indicates the unzipped segment of that particular DNA. Now that segment will then be transcribed into an RNA, and this is our RNA. So this RNA will then undergo several processes, and after that, it will go out of that cell's nucleus through the nuclear pores. This RNA will now be called mRNA since it carries the transcribed message from the double-stranded DNA. Then, this mRNA will be attached to this ribosomal RNA, where the translation process begins. The translation process simply is the translation of the mRNA codons into an amino acid. These are our transfer RNAs. So we have here the free-floating transfer RNAs inside the cytoplasm. So these tRNAs carry with them the complementary anticodons and one amino acid. So for instance, we have this coded or translated codon AUG. It was translated to amino acid methionine, which is our start codon. Then the next RNA codon we have CUC, translated to amino acid leucine. As we go along with this translation process, one amino acid is added or attached to the existing amino acid chain, and as a result, we will create a polypeptide chain known as the protein. This is the process of synthesizing a protein inside the cell cytoplasm. This illustration as well explains the process of translation, segment of mRNA, and this is our ribosomal RNA. We have here the free-floating tRNAs, which carry one amino acid with them. So the translation process begins with the start codon, AUG, and it goes along until the process creates a long polypeptide chain called protein. This activity intends to simulate the steps in translation. So remember that we have two major processes in the protein synthesis. We have transcription and translation. In activity 2C, we just focus on translation. Okay? Again, translation is where the mRNA codes be translated to become an amino acid polypeptide chain. Okay? So the first thing that you have to do is to follow the steps 1, 2, 3 stipulated in activity 2A. Next is to fasten molecule together using a clear tape across base pairs. Now in step 1, you have to modify step 1 since in this activity 2C, you have to follow the color code where phosphate is blue, should be colored blue, ribose sugar should be colored brown, and nitrogenous bases as follows. Now, step 4, cut out the shapes of each nucleotide of RNA. And 5, with your DNA model in front of you, pull apart the DNA model. Remember that you already have a DNA model with you. You need to unzip it, the same with what I showed you a while ago in the process of transcription, where the DNA is unzipped to start the transcription process. Using the right strand of the DNA model in step 3, begin matching complementary RNA nucleotides with the exposed bases on the DNA model to make mRNA. 7, tape the RNA nucleotides. So you should be able to create an mRNA out of that DNA model. Number 8, fasten your molecule using a clear tape. You should use a clear tape. Then imagine that mRNA leaves the cell or the cell's nucleus and moves out of the cell's ribosomes. Meanwhile, transfer RNA is present in the cell cytoplasm. Again, remember that the mRNA leaves the nucleus through the nuclear pores. And also, remember that the cytoplasm is our site of translation where we have plenty of transfer RNA floating around it. Number 9, cut out the two models of tRNA only along solid lines. And number 10, join the tRNA molecules to the mRNA model. So you have to join them. You have to perform complementary base pairing. So you have to pair the mRNA codon with the tRNA anticodon. Okay? When you are finished, tape your model of the translation process on the illustration. Okay, so that's it for your activity 2C titled Relay the Message. At the end, you have to create, I should be able to create or simulate the steps in translation process. At the end, you should be able to, at the end, you should be able to simulate the steps in translation process and create a tRNA complementary base sequence. This is the template which I gave you. You have to cut this out and use the color code for ribose, phosphate, and the nitrogenous bases, uracil, cytosine, and guanine, and adenine. Also, on this part, you have two models of amino acid. This one's the aspartic acid, and this one's the amino acid serine. Since you have this shape which matches with this tRNA model, you need to attach this serine, amino acid serine, to this tRNA model. Also, this aspartic acid should be attached to this tRNA model. Okay? Again, your final output should be a tRNA base sequence with its amino acid chain. That's it, and have a good day.